In this screencast video lecture, we will try to understand about the different types of respiration that has been carried out there in the bacteria. Respiration in bacteria is the biochemical process by which they can able to generate the ATP, that is the energy, mainly by oxidizing substrates like glucose and transferring electron through the electron transport chain to a final electron acceptor, which is usually a oxygen molecule, which on accepting the electron will be converted into water molecule. There are three types of respiration which is commonly carried out there on the bacteria. The first one is an aerobic respiration. It's a kind of a respiration that happens in the presence of oxygen, and here the final electron acceptor is oxygen molecule, which, as I already told, will be converted into water. The key features of this aerobic respiration includes oxygen is essential for this process. During this aerobic respiration, glucose can be completely oxidized or broken down into carbon dioxide and water molecule. Usually before an aerobic respiration, glycolysis will be operating, then Krebs cycle, and finally the various electron carriers generated from these two particular cycle, mainly the NADH and FADH, are all channeled there into the electron transport chain, which finally culminates there as an aerobic respiration. Next, we look at the energy yield during this particular process. Maximum ATP formed is around 36 to 38 ATP per molecule of glucose. This is with respect to prokaryote, it will be little bit reduced. Say for example, 2 ATP will be lower there in the eukaryotic organisms. As I already discussed, the final electron acceptor is oxygen molecule, which on accepting the electron forms into water. Examples of the bacteria which can able to carry out the aerobic respiration includes Pseudomonas aeruginosa, Bacillus subtilis, Mycobacterium tuberculosis. The next one is an anaerobic respiration. Here it's a respiration that occurs mainly in the absence of oxygen. Here, other than oxygen, various inorganic molecules such as a nitrate, sulfate are all acting as an electron acceptor during this respiration process. Here oxygen is not involved. Still here the electron transport chain is been operating, but with different types of terminal electron acceptors. As I already told, it can be of a nitrate, sulfate or carbonate. If nitrate is accepting, it will be converted there into nitrite or nitrogen gas. Sulfate, on accepting the electron, it can be converted into hydrogen sulfide, and carbonate can be converted into methane during the anaerobic respiration process. Compared to that of the aerobic respiration, here the energy yield is lesser, and it also changes with the type of electron acceptor involved in this process. Example of certain anaerobically respiring bacteria includes D-sulfovibrio, which uses sulfate for its anaerobic respiration, Paracoccus denitrificans, which uses nitrate for the anaerobic respiration, and finally methanogens, a group of anaerobic organism which can able to anaerobically respire by using carbon dioxide. That is, carbon dioxide will be converted there into methane during the anaerobic respiration process. The third and the last one is a fermentation. It is again the one which takes place in the absence of oxygen. However, here the electron transport chain is not involved. It is again an anaerobic process wherein organic molecules acts as both electron donors and acceptors. There is no any external electron transport chain operating. However, ATP is produced here mainly by the substrate level phosphorylation process. It occurs without the involvement of oxygen and electron transport chain. Here, during the process of fermentation, glucose is incompletely oxidized. That is, it is not fully converted to carbon dioxide, whereas it forms into various final end products such as organic acids of acetic or formic acid. Sometimes even some neutral products like acetone are formed, and alcohols and gases are again a very typical end products there in an fermentation process. Then the energy yield is very low compared to that of the other aerobic respirations as well as anaerobic respiration. Thus, it will be only a 2 or 3 ATP molecule can be formed per molecule of glucose. As I already told, the final electron acceptor is an organic compound. It can be of an pyruvate or acetaldehyde. Examples of fermentative bacteria are Lactobacillus, which is involved there in the lactic acid fermentation, Saccharomyces cerevisiae, the yeast which is mainly used for beer as well as wine production, is a classical example for alcoholic fermentation. And the third one, Clostridium, is again an anaerobic organism which is carrying out a mixed acid and butanol fermentation, meaning it produces the some alcohol such as a butanol, and some mixed acids include the acetic formic acid. Here is a comparison table which shows the difference between the three different kinds of respiration process.